Let's discuss the second property, that is refractory period. Now the word refractory means not responding. So in the context of the action potential, what do we mean by refractory period? That phase of the action potential during which the tissue does not respond to a second stimulus is called refractory period. Now, refractory period is divided into two types. Absolute refractory period, as the name itself suggests. This is absolute, meaning that even if you increase the strength of the second stimulus, the tissue will not respond. So there is no response to stimulus of any strength. Now, absolute refractory period extends from the firing level, that is the threshold, to 1/3 of repolarization, and its value is 0.5 to 1 millisecond in a nerve. On the other hand, you have relative refractory period, which means that the tissue will respond to a second stimulus provided this stimulus is greater in strength than the first stimulus. Otherwise, it will not respond. So relative refractory period means that the tissue responds to the second stimulus if it is greater than the first. Now this extends from 1/3 of repolarization to the start of after depolarization, and its value is about 1 millisecond in a nerve. So let's understand the concept of refractory period using the graph. Now this is the graph depicting an action potential obtained from the nerve. So you can see that this is the recording of the action potential. Now which part of this action potential falls in the refractory period? So the absolute refractory period extends from the firing level, that is the threshold, which is -55 millivolt, to 1/3 of repolarization. So this duration from threshold to 1/3 of repolarization is the absolute refractory period. Whereas from 1/3 of repolarization to the beginning of after depolarization is the relative refractory period. So that means if you apply a second stimulus in this period, if it is of the same strength as the first stimulus, the tissue will not respond. So this entire duration is refractory. Now this is quite advantageous because if there was no refractory period, then the tissue would respond each time to a second stimulus, and that would cause early fatigue. So this refractory period helps in preventing fatigue. Now why is refractory period so important? As we already saw, it prevents fatigue. Apart from that, it also helps in the separation of individual action potentials. To understand this, let me draw a diagram. So this is an action potential. Now, while this action potential is in progress, let's say you apply a second stimulus, but the tissue does not respond because it is in refractory period. So you have this action potential, you complete it, and then you apply a second stimulus, so you get another action potential. Now in this diagram, it is very clear that there are two individual action potentials. And this is because there is a refractory period which has prevented merging of action potentials. On the other hand, imagine if there was no refractory period, then if this is the first action potential, now let's say you apply a second stimulus here and the tissue responds with yet another action potential. So this is your second action potential. And then there is repolarization. Now in this action potential, there are actually two action potentials merged into one, but we cannot separate them out. It looks as if there is only one action potential. So this confusion is prevented because of the presence of refractory period. The other significance of refractory period is that it enables unidirectional propagation of the action potential. So if this is an axon And let's say there is an action potential here. Now this acts as the stimulus for the adjacent area, and you have another action potential here. Now this action potential would try to stimulate this area as well as again stimulate this area. But fortunately, this area is in refractory period and will not respond. So the action potential has no choice but to move in this direction. So this ensures unidirectional propagation of action potential. Now let's discuss the third property, that is conductivity. We already saw that the magnitude of the action potential is quite large, so it is capable of traveling long distances. This property is referred to as conductivity. Now, conductivity differs between a myelinated and unmyelinated nerve. In the unmyelinated nerve, conduction is electrotonic. What do we mean by this? So this is the axon of an unmyelinated nerve. We apply a stimulus here and there is an action potential here. So you can see that there is a reversal of polarity here. Now because of this, there is a difference in potential between these adjacent areas. So current flows and we refer to this as current sink. So a new action potential is generated here. Now there is a current sink here and that will cause an action potential here. And in this manner, the action potential slowly travels along the axon. Now, the disadvantage of electrotonic conduction is that it is quite slow. On the other hand, in a myelinated nerve, conduction is saltatory. What do we mean by saltatory conduction? So this is the axon of a myelinated nerve. This is the myelin, and in between are the nodes of Ranvier where there is no myelin. Now you know that myelin is an insulator. So what happens is when you apply a stimulus here, there is a reversal of polarity, there is an action potential, but the current sink does not occur between these two adjacent regions because the current cannot flow through the insulation provided by myelin. So there is no choice but for the current to flow from this node of Ranvier to the next node of Ranvier, bypassing this entire myelin sheath. So in other words, the action potential jumps from one node of Ranvier to the next. So obviously conduction is much faster. So the advantage of saltatory conduction is that since the action potential jumps from one node of Ranvier to the next, conduction is much faster. Also, it consumes less energy. Now, many factors influence conductivity, two of the most important factors being diameter of the axon and myelination. We already studied that larger the diameter of the nerve fiber, greater is the conduction velocity. Similarly, there is much faster conduction in myelinated nerves when compared to unmyelinated nerves. Now, even within myelinated nerves, the internodal distance can affect conduction velocity. Now, if you look at this diagram, let's take two myelinated nerves. Now, let's say in this one, these are the nodes of Ranvier. So, they're quite far apart. Whereas, let's imagine another axon where the nodes of Ranvier are very closely spaced. So this is a second myelinated axon. Now, if you look at the first one, the action potential jumps a long distance each time, and therefore conduction is much faster. But here, it is only able to take a small jump, and therefore it would take a much longer time for it to cover the same distance. Therefore, the internodal distance in a myelinated nerve also makes a difference to the conduction velocity. There are other factors, of course, such as temperature, pH, pressure, and oxygenation. Hypoxia can also influence conductivity. So what we learned in this topic was that an excitable tissue can respond in two ways to a stimulus. If the stimulus is subthreshold, then the tissue responds by generating a graded potential, whereas if the stimulus is threshold or above, the tissue generates an action potential. So in this slide, let's summarize the important differences between the graded potential and the action potential. So as we already saw, the graded potential is very small in amplitude, whereas the action potential is large in amplitude. Now, the graded potential does not demonstrate any reversal in polarity, whereas in case of an action potential, there is a reversal of polarity. Briefly, the inside of the cell becomes positive with respect to the outside. The graded potential does not obey all or none law, which means that its amplitude varies depending on the strength of the stimulus. But an action potential obeys all or none law and its magnitude remains constant. Now, a graded potential being too small cannot be conducted for long distances. So it is a local potential, whereas an action potential being large in amplitude can be conducted across long distances. We shall see later that a graded potential can sometimes be inhibitory also. It is not always excitatory, but an action potential is always excitatory and it begins as depolarization followed by repolarization. Finally, a graded potential is initiated by a stimulus, whereas an action potential is initiated by a graded potential. When the graded potential reaches threshold, it gives rise to an action potential. So these are the important differences between the graded potential and the action potential. So with that, we come to the end of this session. Thank you.